Thank you very much, Klara, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, thank you, Kate. It's a pleasure, and I will go ahead and share my screen. Okay. Mhm. Okay, can you confirm that you see this well?
Yes, it's perfect.
Okay, perfect. So, hello everyone, and it's a pleasure to be here. And I'm going to talk about proteins today a lot, also about a few other things, but mainly about proteins and peptides. And a specific question that I would like to address is if we can actually do with less than 20 amino acids. So I first want to give a little bit of background to where this comes from. My group in Prague studies origins of life and protein evolution. So basically, we are interested in how the first proteins could come about from sort of the bottom-up prebiotic environment, but also we are then interested how they evolve nowadays. There are many known and characterized pathways to how proteins can emerge. And in particular, we study the kind of mechanism of proteins arising de novo from previously non-coding DNA. So essentially what some people call random sequences. But what I'm going to talk about today is mainly focus on this first gray area where proteins were likely more simpler, especially in terms of composition, and also how this is possibly tied to origins of translation and later evolution. As I said, we are based in Prague. This is at in the Czech Republic, and actually we are now at the very downtown Prague in this building right here. We moved here about a 1.5 year ago, and it's been great ever since. This is actually not this year's picture from Christmas party. We are yet to hit the Christmas party, and I also wanted to say that. We also have a secondary affiliation to the Institute of Organic Chemistry and Biochemistry in Prague. The work that we do relies heavily on many collaborations, and here's just a list of some of the more long-term co-workers. And our work would probably not be possible without these great people. So, as I said, we're interested in the protein alphabet and how it evolved. This is the protein alphabet as we know it in all life that we know, essentially. And we know from several directions that this alphabet is probably largely unchanged for the last roughly 4 billion years. So from the times of last universal common ancestor. This also means that the alphabet evolved in the roughly 10 to 20% of the Earth's history. And from several lines of evidence, it seems that the alphabet was simpler early on, something that we refer to as the early amino acid alphabet. And essentially, these are the simpler amino acids, those with smaller side chains, those that are just easier to synthesize, and those that were apparently prebiotically available through different routes. For example, these amino acids have been repeatedly detected in carbonaceous meteorites, but they also come up in different versions of abiotic synthesis on Earth. For example, the famous Miller-Urey experiment. So there is quite surprising agreement between these two different lines of evidence pointing to an early amino acid alphabet, which was just composed of the smaller and simpler amino acids. At the same time, we have seen in a lot of these experiments mimicking the prebiotic earth that there were other amino acids, small amino acids similar to those that I just talked about, uh, but those that didn't make it into the protein alphabet as we know it. These are a few examples of these non-canonical amino acids. And if you look at these, it's actually quite a hard task to deduce why these were not selected among the protein alphabet as we know it, and why basically nature employed those late amino acids which had to be probably biosynthesized before they could enter the genetic coding system. So this basically determines the main questions that we have around this project. Why were these amino acids selected from the large prebiotic pool? And, how did this influence the roles that peptides or proteins could play? If there were simple peptides or proteins composed of these smaller amino acids, could they actually deliver some function to early life? Or did they essentially have to wait for evolution of some of the later amino acids? And of course, we are at a seminar Build a Cell, and it's a very obvious question also to ask how did how could early cells look like? How could they work? Could they actually work with just a subset of amino acids, or did cells, in a way that we know them, basically evolve only after some of the later amino acids were here? So these are the questions that I will try to go through today. And I will divide my talk into two areas. The first one will be on reduced alphabets, as the title implied, but I would also then like to talk about some minimal versions of ribosome, which is also, in my view, very kind of important for building cells bottom up. So first, I'll start with the reduced alphabets, and it all goes to the question that I just talked about. Why were certain amino acids selected and others left behind from the prebiotic era, especially? And so to address this question, we recently did a very, very simple experiment. This experiment essentially played with peptides, specifically with 25 mer peptides. Um, to see how their properties would be different if built from these different subsets of amino acids. And for this purpose, we constructed libraries, combinatorial peptide libraries, using solid phase peptide synthesis. In this way, we could really capture a huge area of sequence space because essentially each molecule in our library is unique in its sequence. And just for the clarity, I'm going to show you four libraries that we looked at. So they all have in common is here in the center. These are the early amino acids, those that are canonical. And this blue sector actually makes it to the all 10 early amino acids as we think of them. So this is essentially kind of our baseline. These are the 10 amino acids that we believe are is the subset of our canonical alphabet. Then on the bottom is a very similar library, with the only difference that those branched aliphatic amino acids were substituted with their unbranched variants. So those side chains, which chemically look very similar, is just that they are unbranched. They're very linear. Norvaline, norleucine, and 2-aminobutyric acid instead of isoleucine, leucine, and valine. And then on the right side, we play a very similar game, but this game is focused on positive charge. Something that I didn't point out before is that those 10 early canonical amino acids include only acidic residues. They don't contain any basic amino acids, which would be arginine, lysine, and histidine in today's alphabet, that those three seem to be prebiotically quite hard to synthesize. But there are some alternatives which might be prebiotically available, such as diaminobutyric acid. So what we are trying to address by this right side is, okay, if we have a 10 early plus arginine library versus 10 early plus a prebiotically available diaminobutyric acid. And what we did in this project is actually very simple, as we just compared the physical chemical properties of libraries just like these four. And we first looked at solubility of these libraries. And on top is the solubility across different pHs for the canonical libraries 10E and 11R, the one with arginine. And on the bottom are the non-canonical counterparts to these libraries. And there is a very clear message from this plot showing us that the non-canonical versions of our libraries, those that contain some non-canonical variants, are actually more soluble than the canonical ones, which you might think is quite good, right? Solubility is something that we want to see, especially with proteins. But what we have also learned over time, especially by breaking these random sequences, is that solubility is actually another sign of the coin of secondary structure content. And this is what we saw with the next experiment when we did circular dichroism of these libraries. And we see that the top ones actually have more secondary structure content. This is something that is obvious from these like minima or shoulders, somewhere between 200 and 220 nanometers. They have more depth, these two libraries, than those non-canonical variants. So while the non-canonical variants are more soluble, that actually also corresponds to less secondary structure content. So we think that this was actually probably an important factor during the selection of the alphabet, that those amino acids that were more important for delivering structure in proteins were retained, while those alternatives, which would not be so good for building structures, were purged or not selected. And in particular, this 10E library, those amino acids that we actually believe were prebiotically available from the canonical set seem to stand out in many properties, not just within these four libraries, but in among about 15 different libraries that we have tested so far. One of the things that we observe that is quite striking about this library is that it actually tends to form assembly of these random peptides. This is shown here. This may be a little bit harder to follow, but nothing scary here. So what's happening is we are looking at the size exclusion chromatography of these libraries. This basically shows us the species that are found in these peptide libraries. And in all of them, you see a peak around 15 milliliters on the column, which basically shows the monomeric species within these libraries, just individual peptides, different pHs. But what we also see for the 10E library is a small peak around 8 milliliters, which is something that is higher in mass, a species that is higher in mass, and it's especially from neutral pH up. So we decided to look at this in a little bit more detail. I mean, this is something if you were purifying a protein, you wouldn't be paying much attention. But if you are interested in origins of life, and especially in origins of protein, peptide assembly is actually very interesting phenomenon. So we decided to focus on this a little bit more. And here is actually just the enlarged version of this chromatogram. And if we look specifically at this small peak around the 8 milliliters, and we look again at CD spectroscopy, we actually see something very exciting. This may not be super exciting for you if you're not used to looking at CD spectra, but this is essentially what textbook beta sheet spectrum looks like. I have to say I've never seen anything like this before until these results. And what it essentially shows is that our assembly is held together via beta sheet peptides. Um, this is something that might remind some people of amyloids. On the other hand, we have to remember that our sequences are not identical one to each other as you will see them in amyloids. And they're also quite different in composition than most proteins appearing in amyloids would be. So we looked at the electron tomography, and we also did not observe any fibrils, which would be very typical of amyloids. What we have here are rather some globular assemblies. Which is kind of the exciting part because if you just work with peptides, especially during life's origins, where you still don't have any translation at work, which could give you protein length molecules, globular assemblies are quite exciting because it means your peptides are coming close together, which can create environments for catalysis or for binding, basically concentrating some things together. So yeah, just to summarize, uh, for getting too excited, what we observed in this part of the study was that all the different periodically available amino acids, which were non-canonical, which didn't end up being selected in our alphabet, actually decreased secondary structure propensity. The 10 early amino acids actually somehow stand out and among all of these libraries that we studied and among the different amino acids in a way that they form these higher assemblies. This is something that we have not observed with other alphabets, with differently charged alphabets. This is something that is very sort of intrinsic to this overall acidic step. And we tend to think of them as possible stepping stones to protein-like structures. On the other hand, something that we did not see, and this part of the project was, could early proteins actually be composed of these 10 early amino acids? Basically meaning if there was already translation developed at the stage of this 10 early amino acids, could it actually make proteins? Could it actually make structures like those that we know from today's biology? And this is something that we addressed recently using a very different sort of way of thinking. So we're kind of shifting away from combinatorial libraries here. And we decided to apply the novel tools of protein design in collaboration with Ingemar Andre from Lund University. And so we basically asked whether some of the most ancient folds could be reconstructed using this half of the alphabet, the 10 early, what we think early amino acids. And the way we did it, we basically nominated those folds, which are considered to be among the latest ones, fixed their backbone coordinates using our diffusion, and then populated these folds, these coordinates using protein MPNN. This step was, of course, the most important one because we had to penalize all those amino acids which we did not want to populate our protein structures. And I'm not going to go into detail, but just show you three folds that we successfully redesigned. These are actually NMR structures. The green ones are NMR structures of our 10 early variants of these folds overlaid with the original red wild-type structures. This story is now on bioRxiv, so you can chase it if you're interested in more detail. But I'm just using it here to basically deliver the point that proteins, as we know them, simple folds, could actually be constructed using only early amino acids. And I'm saying this specifically here because I think this can be sort of very minimal approach to building simple cells. And of course, there is this caveat in our way of thinking here that we don't really know when translation evolved, when ribosomal synthesis evolved, whether this was before the kind of evolution of the amino acid alphabet. Already there could be a primitive sort of version of ribosomal synthesis with these early amino acids existent. Which directly takes me to the second part of my talk. We've been talking approximately 20 minutes, so I think this will work fine. And I'm just going to tell you sort of a little story that we are currently working on towards the question. So I'm sure you noticed in the last few years that there's been a lot of attention given to the very center of the ribosome called the peptidyl transferase reaction. This is the part of the ribosome, the very central part of the large subunit where the peptidyl transferase reaction happens. So that place where amino acids get connected to one another and form a peptide chain and a protein. Two groups independently reported that when they extract this part of the ribosome, the peptidyl transferase center, out of the ribosome and work with just this construct, that they see the PTC reaction happening. And of course, kind of connected with the whole RNA world scenario. Quite interestingly, about a year ago, there was a follow-up article actually comparing these reactions, those by PTC only, which here would be the green, the green bars, with the whole ribosome. That's essentially the positive control, but also with a mock scenario. So reaction where there would be no RNA, PTC RNA present. And quite surprisingly, in this study, the mock reactions actually came out more efficient, more effective than those reactions where the PTC RNA was present. Basically raising the question whether RNA actually is the catalyst here, and to what extent, does the reaction happen between does the reaction happen kind of spontaneously? So this is something that really got us interested because, as I told you, we are interested in proteins and in peptides. And so we were very motivated to look at this structure of the PTC and kind of review it, especially in the context of the ribosome as we know it today. And here, the PTC as the blue species is actually highlighted in the center of the ribosome, but also kind of shown in the context of the protein material around it. So these brown species are the peptides, the fragments of proteins that are actually found in the proximity of PTC. And we have seen many examples of how RNA and protein have co-evolved in nature. And so we were very interested in how could peptides actually change the structural and functional properties of this important peptidyl transferase center that is now sort of surrounded by many interesting questions, especially regarding the catalytic potential. And so we just started playing this as a bit of a side project. And one of the first things that we observed was that when we mix these two species together, we actually see coacerates, formation of coacerates or droplets or liquid-liquid phase separation. And we published this last year. And I'm not showing this here, but we also confirmed that both the RNA and peptides actually localize in these coacerates. This is currently a lot of work in progress, but of course we are interested first in how do the peptides actually influence the properties of these droplets, and especially when it comes to RNA activity, because activity of ribosomes has been previously sort of tested under different conditions in these droplets. And the role of peptides or magnesium, or especially their kind of a balance has been reported very important for ribozyme activity. So this is something I'm not going to tell you yet. I don't know, we don't know, but something we're heavily working on. We are also interested in on the peptide side, how much is this dependent on the canonical full alphabet, or whether we can possibly see the coagulation and the activity happening with um, um, peptide content that would be more sort of similar to the prebiotic composition. We already have a sense that it's very hard to do without positive charge in the peptides, but at the same time, we see that the sort of earlier alternatives of positively charged residues might be sufficient to deliver the effects that we previously observed. So this is something in progress at the moment. We think we kind of stumbled upon this serendipitously, but we think that early translation could actually co-evolve in environments similar to coagulates. It is still unclear how peptides or protein content would influence it. This is something we yet would like to see. But of course, we think it's important to consider minimal ribosomes because the ribosomes, as we know them, are very kind of a huge complexes of molecules. And for building cells bottom up, it might actually be really convenient to have some minimal versions of these molecules. So that's again the reason why I'm talking about them here. And finally, this has of course been work of many people that I haven't mentioned on the way, most of them from our group at Charles University, but also this was from collaboration with Kosuke Fujishima at LC, Giuliano Zanchetta at University of Milan, Ingemar Andre, who I mentioned at Lund University, and Stephen Fried at Johns Hopkins. And something I wanted to mention was that we are currently looking for one or two postdocs that would actually be working on something different than I discussed here, but something very related to synthetic cells and minimal cells. So if you would be interested, please let us know. And I think this is it from me. 30 minutes. So I hope that is okay, as promised.
Thank you. Thank you so much. That was amazing. There is quite a bit of questions in chat. Can you see chat?
Okay, hold on. I'll stop sharing and I can call the presentation back if needed and just to make the chat bigger here. Okay,
If
I
you
do
could read
see.
a question out
Yes.
loud, because people watching the recording won't see the questions.
Okay, I'll just start from the earliest ones. So the first question was from Evelina Bird. When people assemble minimal alphabets, do you always just test minimal cell set of canonical amino acids, or do you ever try some of the non-canonical amino acids, ones that didn't make it into protein? So yes, that definitely something that we tried. Of course, something that we did not do was having alphabets that would be entirely of non-canonical amino acids at this stage. This is something we are also interested in and working on a little bit, but all those non-canonical versions were kind of mixes of canonical with additions of non-canonical amino acids. And what we saw, as I showed, but hopefully managed to deliver, was that they usually decrease the secondary structure potential. They don't seem to be so good for selection into protein alphabets. So I hope that answers it. If you have a follow-up question, I'm happy to talk about it. So I'll go to the second one, if that's okay. That's Basak, University of Minnesota. Thank you. So I was wondering if you have tried using fewer than 10 amino acids to form functional proteins/peptides. So Not yet. I mean, this is a very, very interesting area of research, and we would like to go this way. On the other way, you are from University of Minnesota. You might know of the work of Burkhard Selig, who I believe are working with even less amino acids. I believe with as little as 4 or 5. There is a paper from them from roughly 2000, 1917, something like that, from Matilda Newton as the first author, where they showed some properties of sequences made of a smaller number of amino acids. So I believe this is something that we should be kind of looking into, and I believe there is work being done in this direction from our colleagues. Okay, there's another question from Heather. Thank you. Do you think it's possible to figure out the order of amino acids coming to biology by looking at the evolution of tRNA, which isoacceptors are older? Yes, this is a really exciting question, I think. Um, so of course we would be very interested in the order of how amino acids became incorporated into the alphabet. This has been approached by some people, not just by looking at evolution of tRNA, because there are many, different possible kind of perspectives on this problem. And I don't believe we have the resolution at this moment. I think that kind of there is a little bit of sort of discrepancy between the different methods addressing this. So there is a recent study kind of inspired in ancestral sequence reconstruction of proteins that have aimed kind of reconstructing this order, and it's in a little bit of clash with some of the earlier work trying to address this from the sort of
Need some tools. Hmm.
prebiotic chemistry point of view. And so I don't think we have the resolution at the moment from these different directions, and we're rather kind of left with looking for how these methods, where they meet, how they agree. And I do actually think, I do agree with you that having a way to kind of study the deep evolution of the whole translation apparatus, not just tRNAs, but amino acid tRNA synthetases, I do believe that we could hopefully derive to more clarity. I don't think we will ever be able to say which was number 11, 12, 13, 14, 15, and so on, because it's quite possible that this also didn't work like that at the moment, that very rarely I could imagine that the evolution was not clear like this, that there were also maybe some periods when the whole system, the coding system was likely more permissive, more promiscuous. And so it's a little bit kind of blurry. And I don't know whether we will ever have tools to address this blurry period of translation evolution. So I think it's a great question, though, and I think possibly more could be done than we know today, and I hope it will come. Be interesting. Okay, there's a question from Claudia Adair. Do you ever look at lipids interaction with simple peptides and lipids? Uh, we did attempt a little bit, not with lipids as we know them, but with fatty acid vesicles. This is something that we have on bioRxiv. I did not talk about, maybe I should have. So we did try actually looking at some of the peptide libraries that I talked about, those with canonical but also non-canonical amino acids, and see how they affect formation of vesicles and also how they interact with fatty acids. What we saw was that there are differences between different peptides, between different peptide compositions. We saw that while the early amino acids, peptides from early amino acids are quite capable of somehow interacting with vesicles, things become a lot kind of more efficient when there is positive charge around. This positive charge can be also from the non-canonical variants, so diaminobutyric or diaminopropionic, but there is definitely improvement with the canonical versions of positive charge, arginine, lysine, which if interaction with lipids was one of the factors kind of affecting the evolution of the coding, it would make sense again that arginine and lysine would be selected over those early possible variants. And to me, this also is again another kind of factor telling us, that the early evolution could probably, the early alphabet could probably do something on the peptide protein level, but to really get to cells as we know them, we think that some positive charge was really needed in peptides and proteins and would come with significant advantages, adaptive properties, I would say. So again, great question, and thank you. Okay, and there is a really good question, last one from Henry Taylor. Can secondary structures of simplest peptides be stabilized by iron 2 plus that was available before oxygenation? So that's a great question. I don't think I have the answer. There was there were some articles kind of proposing that this would be the case not just for iron 2, but also possibly for other divalent metal ions. We tried introducing divalent metal ions into the study of our assemblies, of those peptide assemblies that I talked about. And we definitely see that there is something happening. We see that our peptide libraries undergo some change with metal ions. They seem definitely to be binding it. They seem to be competing for the binding with EDTA, for example. But we don't yet understand what's happening with the structures, with the secondary structure as such, because there also seems to be kind of increase in this assembly propensity when this happens. So probably these metal ions might be helping to drive the assembly, maybe more of that than just change the secondary structures themselves. But this is definitely something that I believe would be very good to look at on specific examples of peptides where we could actually kind of mechanistically study what's happening and be able to see specific secondary structures. Ideally, I would say using NMR, something higher resolution than just a CD spectroscopy. So yeah, thank you for the question. I think it's a really interesting sort of idea to look at, not just the metal ion binding, but for us also binding to different organic cofactors. As we know from today's biology, how about half of the proteins as we know them are actually dependent on metal ion or organic cofactors for their catalytic activity. So I think this is a very important question. And again, I think it's super relevant for thinking about, building cells bottom up. Like, what do we actually need? What should be our ingredients? And I think all of this is definitely on the list. I guess Kate would agree.
I absolutely agree. And this sounds like a fascinating. There's basically enough directions for research in this last 10 minutes when you were asking questions that it's going to keep us employed for the rest of our lives. Hopefully.
Yeah, I think so too. Yeah, I think so too. So thank you. I think this was the last question from this list.
Yep, that's the last one I see too. And if there are no more questions, I just want to say thank you so much again, Klara. It was fantastic talk.
Thank you.
And
it was
everyone.
a pleasure. Yeah, thank you.
And have a great rest of your Monday.
Thank you.
Bye.
You too. Bye.